Welcome everyone to the BIMR seminar. I'd like to introduce Professor Samantha Payne. So this one, that's biomaterials background, but a lot of the biology side, but it's important to still hear these talks because biomaterials, or everything we make has to interface with biology at the end of the day, right? A lot of times we make stuff and never do the final steps, and let's see if it actually works. And Samantha's good at doing all the interconnections and knows the biology much better than I do. She started her life at Guelph for undergrad and master's, went to PhD in Molly Shoyket's lab, and that's how I know her, even though we didn't overlap. She's a lot younger than I am. I was out of the lab by the time she joined. And went to do her postdoc at Tufts, where she really got into the biology and the signaling stuff and really measuring what cells are doing in different environments. And now she collaborates with Payne in the BIMR community and uses the iFlow puts. And I think we'll hear a little bit of that today, right? Mm-hmm. So she's taking technology developed here and actually using it for scientific aid. All this thing, right? So with that, welcome Samantha, and listen to her talk.
Thank you. Thanks very much, Ryan. Thank you so much for inviting me here today. It's a pleasure. And yeah, we'll see if we get scientific gain. Still kind of in the works. So yeah, I'm going to talk to you today about a couple of stories. I've kind of kept it more biomaterials or at least device focused given the audience, but I am really a biologist that kind of masquerades when I want to and when it's convenient for me as an engineer. So, but please feel free to ask any questions along the way. And so yes, I'm at the University of Guelph and I'm in the Department of Biomedical Sciences. So we're part of the Ontario Veterinary College, which is kind of a unique place to be. We've got a lot of cool animal models, some larger animal models, things like that. If you ever want to take advantage of that kind of thing, you can always let me know. Okay, so I'm going to give you a little bit of background in the problems that we're looking at addressing in the lab, and then I'll talk to you about how we are addressing those problems. And I might actually, sorry, I didn't bring a laser pointer. I might at least put one on here. So one of the problems that we're looking at in the lab is skin repair. So when you have an injury to the skin, you'll get a scar forming, and that's sort of the default response. So in this little schematic here, you undergo a period of inflammation, immune response, immune cell infiltration. Then you'll have the main cells of the skin, keratinocytes, which you're going to hear me talk a lot about today, will become activated. They will migrate and proliferate, fill in the wound and re-epithelialize it, so close up the skin. And then just deep to that, you get collagen deposition by dermal fibroblasts, which you'll also hear me talk about today, and that's what forms the scar. So scars are fine. They kind of, seal over the wound. They prevent infections and things like that, but you're gonna have a loss of function and a loss of normal tissue morphology. And then on top of that, another kind of option as a sort of disordered response to injury is that you can have prolonged inflammation, prolonged immune response, and then you can have something like a chronic non-healing wound. So you might be familiar with the idea of like an ulcer or some sort of wound where it's not closing up, it's sort of chronically inflamed. And that obviously comes with some challenges. Just to throw up a couple of stats here. So of course, the function of the tissue is going to be compromised. The barrier function of your skin is compromised. So you may be susceptible to infection, aesthetic concerns. And so just a couple of numbers here. The scar therapy market in the billions of dollars in 2023, as well as just even treatment for wounds, both acute and chronic. So it's a large problem. So I just told you about the normal kind of default healing response in mammals, but it turns out that a lot of other mammals actually can fully regenerate rather than form a scar. And so this is of interest to regenerative medicine researchers such as myself. So we see this kind of range in response across species. So I just talked about us mammals, and of course we use mice a lot in the lab. We're kind of limited to cell renewal after injury or the scarring that I talked about in the skin. But as you look across the animal kingdom, you get different responses. So I have a colleague that works on leopard geckos that we collaborate with, and they're able to regenerate their tails and pieces of their heart and their skin. We look at amphibians, such as newts, salamanders, as well as axolotls. They can regenerate whole body parts. So you can amputate a limb and they'll grow back almost a perfect replica. And then as we look kind of further away from mammals, I guess kind of evolutionarily, we start looking at fish. So zebrafish can regenerate a lot of different body parts. And then we have nematodes, which you can cut in half and they'll regenerate the entire other half. So there's this huge range, and that was really interesting to me because it sort of, raises the question of like, well, if these animals can regenerate, why can't we as mammals? And so that's sort of a lot of the foundation of what I'm interested in as a researcher. And so why is this response different? What causes this difference in response to injury? And of course, as you can imagine, there's a lot going on in a tissue after injury. So there's a lot of microenvironmental factors that are going to control the cell response to injury. So this diagram here is just sort of like a summary of some of these factors. So you have your cell in the center here. And so of course, you're going to have things like soluble factors, growth factors, nutrients, things like that, that are going to bind to receptors and cause signaling cascades. You might have something like physical cues. It could be electrical, they could be thermal, mechanical cues. Some of you are probably familiar with that if you're in the biomaterial world as a tunable property to get the cell to behave how you want it to. The extracellular matrix, so everything in a tissue that isn't cells, of course, again, also has a lot of different signaling factors, so biochemical, biophysical, stiffness, composition, things like that. And then lastly, there's neighboring cells. So there's very often multiple cell types in any given tissue. The skin is no exception. And so we're really interested in my lab in nerve cells. So what are nerves doing and what is their role in tissue repair? So, and we're specifically interested in the peripheral nervous system. So we have, of course, we can break up the nervous system into two parts. We have the central nervous system, that's your brain and spinal cord. And the peripheral nervous system is basically all the other nerves in your body. And most tissues of the body are highly innervated, or at least somewhat innervated. So they're really ubiquitous. They're often present in a tissue. And so that sort of begs the question is, what are they doing there other than obviously sending signals back and forth from the brain to the body? And I'm going to talk to you today kind of specifically about sensory neurons. So this is just a schematic showing you kind of the pathway from the brain to the tissue. And of course, you've got your the sensory side of things in blue here. That would be your dorsal root ganglion. So those are the collection of sensory cell bodies, neuron bodies, and they'll be sending out receptors to the skin. And so that's sort of what the focus of our work in the lab is on, these dorsal root ganglion nerves. And so it's been observed for a number of years by other people in other labs that nerves are really important for tissue repair and wound healing. So this is an example here from a paper in 2022 from another lab, and they did kind of like a simple, this is on mice, and what they did was they reduced the innervation to this area of the skin and then compared it to normal innervation, and then they did a biopsy punch in the skin, and they looked at healing over the course of 17 days or so. And so what you can hopefully appreciate here is that normally you get pretty good wound healing response here in the skin in the normal mouse. But if you reduce the innervation to that area of the skin, it doesn't really heal. So it's a pretty dramatic effect. If you don't have the nerves there, you won't get proper healing. And I'm not really going to talk about this in this lecture, or in this seminar, but I think it's just like a really cool and dramatic example of this to further illustrate the point. I touched on appendage regeneration, so things like axolotls, if you amputate their limbs, they will grow back a near perfect replica. And this is also very dependent on innervation. So this is from a paper, kind of their schematic here, showing you got the innervation of the limb, you amputate, it grows back. If you denervate, so if you remove the nerve supply to that limb before you amputate, you get no regeneration. So it's a really dramatic effect. So just the nerves not being there, you won't get regeneration. And this is really cool. I just like this image. I think it's neat. Kind of if you do the reverse experiment. So in this case, what they did was they grafted a little piece of skin from another area of the body onto a limb of an axolotl. And then they rerouted a nerve to that little piece of skin that they grafted. And when you do that, you can actually induce a limb to form. So they call this the accessory limb. So there's the original limb here in A, and you can see this nice bone stain here. So when you actually redirect a nerve, you can get a pretty good looking limb that will grow in that place. So that suggests that the nerves are directing this process in a way that's sort of not really fully understood still. So I touched a little bit on this, how we have different species that have different responses to injury. And then, of course, there's a number of model systems that we can use to study tissue repair. So I kind of mentioned these non-mammalian species. They're spontaneous regenerators. So you don't have to manipulate them to get them to regenerate. You can study the process. But it's getting better, but there's a lack of kind of molecular toolkits that you can use in some of these species. And then, of course, they're phylogenetically distant from humans, right? They're less closely related to us. And so if we want to translate these things, it's maybe a little bit a harder argument. Then, of course, we have mammalian species. However, we mostly don't have instances of regeneration, so we're mostly scarring. But we've got a lot of molecular tools, and these rodent models, which are commonly used, are a little bit closer, evolutionarily speaking, to us. But then there's sort of a third category of system, and that's what I'm going to talk to you about today, and that is in vitro models. So models of cell culture. And so these offer the advantage where you can isolate different cells and factors, so very specifically testing certain aspects of that larger microenvironment. Maybe easier to manipulate than an animal model, but then you kind of have that translation limitation where, again, you kind of probably still are going to have to, down the line, go into an animal model. But definitely some advantages there. And so in my lab at Guelph, we're kind of a mix of all of those things to some extent. And so the idea here is in our lab is that we'll take in vitro models of tissue repair and regeneration, animal models, kind of back and forth findings from each, translating them, and then ultimately to translate into like a therapeutic of some sort. So on the culture side, and what I'm going to talk about mostly today, we're kind of essentially looking at the interaction of peripheral sensory neurons with various cell types that we know are involved in tissue repair. So keratinocytes, like I mentioned, skin cells, fibroblasts, which are these kind of connective tissue cells that are found in a lot of different tissues, and they're pretty important. And then stem and progenitor cells as well. So the interaction of the nerves with these cell types. And then I'm not going to talk about it in this seminar today, but we also have some animal models of regeneration, so digit tip amputation and skin wound healing. So the first story I want to talk about is our work that it was published late last year on peripheral sensory neurons and fibroblast interactions. So I've generated a fancy QR code there. If you want to take a look at it, that was published in ACS Biomaterial Science and Engineering just late last year. So I'm excited to kind of share that with you today. I don't know if anyone, I see a couple, okay. So I talked to you a little bit up front about the process of skin wound healing and scarring. I'll just go into a little bit more detail that's relevant to this project. So in the skin, in the dermis, you have your epidermis here, which is this purple layer of cells in the image. Below that, you have your dermis, and that's going to be populated by dermal fibroblasts, these connective tissue cells. And so when you have a wound in the skin that's going to, cut through both the epidermis and the dermis, one of the steps that's involved in wound healing is activation of these dermal fibroblasts. They'll go from a quiescent state and become activated. And so activated is sort of defined as their morphology changes. So they go from like a spindle shape to more stellate shape. They'll secrete collagen 1, and that's the main component of the scar tissue. They will proliferate, and they also express a few different markers, including alpha smooth muscle actin, alpha SMA there. So that's sort of how we characterize when a fibroblast has become activated. And so that step is necessary for skin healing. And it's sort of a balance. You want activation to deposit the scar tissue to heal the wound up, but you don't want, prolonged activation where then you get something like a keloid scar, which is sort of like a bigger, larger scar. So there's a balance there between the quiescent and activation of the fibroblast. And so this project was led by a former master's student of mine, Solsa Kariba, who's actually now here at Mac doing a PhD. Couldn't convince him to stay. I actually lose a lot of people to Mac. I will say that. I have master's students at Guelph, and they say, oh, I want to try somewhere else. And then they go down the road to Hamilton. And I'm like, okay, that's fine. You guys are great, so what can I say? So yeah, I'm going to tell you about this project here that Solsa completed. And so then the question is, do peripheral nerves play a role in fibroblast activation? So we know they're becoming activated. Of course, there's a lot of things going on in the microenvironment, but we suspect that the nerves are playing a role, so we wanted to investigate that. So for everything I'm kind of talking about, the source of the nerves that we use for these studies are dorsal root ganglia, DRGs, so you'll see that abbreviation. And this is just showing you how we actually get these neurons. So we'll take adult C57 black 6 mice, and then we actually will harvest the DRGs from the spine, kind of clean them off, digest them a little bit, and then we seed them as whole DRGs. So these are kind of whole nerves that are put in the dish. So they're going to contain neurons, but also some other cell types as well. And so this project was based on cell culture models. And so just to give you kind of like a slight overview of different approaches that we look at in the lab for co-culture. So when I say co-culture, that's kind of culturing two different cell types together in some way. And there's different approaches for that. I kind of have the two main ones here. So you can, of course, do like a direct co-culture where you add these two cell types into one dish together. And that's nice because you can kind of test the direct contact of these cell types potentially, as well as the effect of secreted factors. However, you might have incompatibility issues with the media. So especially neurons and nerves are quite sensitive to how they're grown. And so if you have a more sensitive cell type, you might not be able to use a different media composition. And so you can run into issues with trying to figure out what's the best media to grow both of these cell types in. So I guess kind of a solution to that, or one thing you can do instead is do something called conditioned medium. So you'll hear me talk about that as well. And so that's where you're not going to have direct contact, but what you're going to do in our case is we grow the nerves in one dish. We allow them kind of to grow a while, and then we harvest that media. So that's the conditioned media. And then you can add that to your other cell type and see the effect essentially of the secreted or soluble factors. And so you're not going to have direct contact between the two cell types. So both of these kind of have their pros and cons. And then we came up with kind of like a third, I guess, option for this co-culture. And that's where Dr. Boyang Zhang comes in of McMaster University. And so I contacted Boyang a couple of years ago, and I said, I'm looking for co-culture devices. And he said, well, we have these ones that we developed called the iFlow Plate 384. So that's what I'm showing here. And so it's sort of this, it's in a kind of modified cell culture plate, standard size. And then it's got essentially it's triplicates of wells. So you can see it nicely here with the different colored dyes. And these three wells are connected by small channels, you can see here. And there's kind of what it looks like in a 3D look. And so we thought, okay, well, if we use these, what we can do is actually kind of segregate out the nerves from the other from the fibroblasts, and we can culture them in their own media with kind of minimal mixing, but we'll still have contact because what you can have is the nerves, the neurites growing from the nerves to connect the fibroblasts. So this schematic here is showing how we set this up. And so again, you've got kind of your three wells connected by these little channels. So we can seed in our nerve, our DRG, into the one channel here, add media, and then you kind of wait a while, and then you get it extending axons, the neurites, from the well that you seed it in into the adjacent well, so through the channel here. And then once you have that, what you can actually do, and you can kind of see the different media, they're colored differently in this image, is you can then seed in whatever cells you like. In our case, we seeded in human dermal fibroblasts, so HDF, it's labeled here. And so that you end up with this kind of co-culture system where you can somewhat stratify the media, but you have direct cell contact and soluble factors if the nerves are secreting factors into that fibroblast chamber. So it's sort of the best of both worlds from the direct and indirect co-culture. So we optimized that. And essentially what we did was, again, we harvested the DRGs, the nerves from mice, seeded them in the chamber here on the left them for 14 days to extend axons. So we gave them lots of time to extend out axons, seeded in the dermal fibroblasts into the center chamber, and then we co-cultured them for another 7 days and essentially assayed changes in the fibroblast phenotype and kind of behavior and function. Those functions that are related to wound healing, so collagen 1, proliferation, changes in morphology, and the smooth muscle actin expression. So here is what one of them looks like. So from the paper here, so this is what it actually looks like when it's here in the iFlow plate device. So we can see here, this is our DRG chamber. This is immunostained with beta-3 tubulin, which stains all neurons. So you can see the nice axons projecting from the DRG body. So this is where the cell body is located and all the axons. And you can see the axons extend through the channel into the adjacent chamber where we seeded our fibroblasts, and they were labeled with something called cell tracker, just so we could see them. So they're in red. So that's how that actually looks. And we were able to get DRG survival for up to 21 days, and probably we could go longer. So it was a pretty good system. We got the DRGs living, sprouting for the most part. So we were pretty happy with that. So then we went ahead and looked at the fibroblast response. And long story short, the co-culture with the DRG, with the nerve, increased fibroblast activation. So this is just showing you, so we did a lot of immunostaining for this. And in this one here, we've got just our fibroblasts alone on the top row, and then fibroblasts with the DRG nerve, stained for Ki67, which is for proliferating cells, and then collagen 1 to see how much collagen 1 we had. And then we quantified that. And so we found both for proliferation and collagen 1, there was an increase when we had that nerve present in the adjacent chamber. So when fibroblasts were co-cultured with nerves, it increased their proliferation in collagen 1. And then, maybe not a surprise since I told you it increased activation, we also saw increase in alpha smooth muscle actin expression with co-culture with the nerves, which you can see here, staining example and the quantification. And then we also kind of looked at the cell size. So we looked at a few metrics. We used phalloidin, which is a cytoskeletal dye, so you can kind of measure the area, perimeter of the cell, eccentricity, a few other components. And we found that both the kind of perimeter of these cells and their area of the fibroblasts was increased when they were cultured with nerves. So they seemed to be, the cells were a little bit bigger. We're not entirely sure what that means. We were kind of looking for maybe that change in like the stellate morphology. But so there did seem to be sort of an effect on the morphology of the cells, at least in terms of size, when they were cultured with the DRGs. And so we thought that was pretty cool. It was pretty exciting that the nerves do seem to be having an effect on the fibroblast function and their activation. And then so what we also wanted to test was whether or not this required direct contact. So that kind of like neurites sprouting through the iFlow plate so that they're kind of connecting to the fibroblasts. So we did a separate set of experiments where we did that conditioned media. So we grew DRGs, took out the media, added it to fibroblasts, same length of time as to kind of match our iFlow plate experiments. And then we measured, again, some of those activation parameters. So again, the Ki67 for proliferation, collagen 1 expression, and we found there wasn't any significance there. So it seemed like our setup in the iFlow plate was able to promote fibroblast activation, and we didn't see that same promotion when we just used conditioned media from the DRGs. So there's more than one reason why that could be, but it did seem kind of like there needs to be more of a direct contact than what you would get with just adding conditioned media. So the last thing we kind of did was, because the iFlow plate has so many chambers in one plate, is we kind of wanted to see if we could put that high throughput capability to good use. And so something else we measured was cytokine secretion by the fibroblasts. And so we were able to kind of, because of the setup of the iFlow plate, each of these A, B, C, and D would be a mouse, so like a biological replicate. So we were able to harvest DRGs from different mice simultaneously, have them all in one place, so to kind of run replicates, all at once with our controls there. So again, it'd be the DRGs with your human dermal fibroblasts or the human dermal fibroblasts alone. And then we could just culture and then sample the media from the HDF and take a look at, we just used commercially available cytokine analysis. And so we chose a panel of kind of angiogenic and growth factors that are known to be important in wound healing, amongst other things. So this is just showing you the list of them there. And so our control was just a blank media control with no cells in it. the fibroblasts alone, and then the fibroblasts with the DRGs. And we found it was there was some variability, but we did find it seemed to be that when you have the DRGs, there was an increase in secretion of some of these proteins. So this graph here is the same data, just sort of plotted differently. So it's the HDF DRG against the fibroblasts alone. So we saw a fold change here in some of these proteins, including things like EGF and HGF, which are growth factors that are known to be involved in skin wound healing. So that was kind of nice to see, and just sort of showed, again, the capabilities of the iFlow plate to have this high throughput capability that a lot of co-culture devices don't have. A lot of them are just sort of like, 2 or 4 chambers in one device. So it's kind of a nice use of it that way. So that was kind of the bulk of what we have done so far. And now we're starting in the lab, and I won't be able to tell you about it today because it's in the works, but we're starting to look more at the mechanism of the control here. So we've sort of demonstrated that having the nerves does seem to activate the fibroblasts. So the next step is then how are they doing that? What's the mechanism? There's a few kind of hypotheses out there. The nerves might be affecting the extracellular matrix, so they can actually change protease expression, so MMPs, which will affect the extracellular matrix. It could just be through kind of like different ligands signaling, so growth factors, like I mentioned. Also inflammatory signals. So there's a few kind of routes that we're now exploring to kind of get to the bottom of the mechanism here on that. And then the other aspect of that I'm really excited about is in collaboration with the Zhang lab, we're now looking at trying to build a 3D kind of version of the model. So very few co-culture devices actually consider innervation as a variable, so it's often lacking in these models of tissues and organs, but they're innervated in the body, so I think it's an important component. So we're working towards now kind of translating our 2D model of the DRG growing on the bottom into a 3D hydrogel. So I think it's going to be pretty exciting, probably a lot of challenges to get the neurites to grow into a hydrogel, but we'll see where that goes, so stay tuned for that. So again, just bringing it back here, I talked about our work on a project looking at sensory nerves with the dermal fibroblasts. And so now the second half of my talk, I want to talk about a separate project that we just recently had accepted for publication, looking at the interaction of the sensory neurons with keratinocytes. So those skin cells. So you can look, it's kind of like an ugly proof still, I think, when you go to this link, but you can take a look at that, and hopefully we'll have the full version up soon. And so a similar kind of idea, knowing that nerves are important for wound healing, what's the actual cell mechanism that's occurring here? Like, how on a cellular level are the neurons affecting keratinocyte behavior? So I'll just talk a little bit about wound healing again, bring your minds back to that. So when you have a wound in the skin, now we're looking at the epidermis here, so that's mainly composed of keratinocytes. And so you can see your dermis here below. Epidermis are these layers of keratinocytes, and as you kind of go from a basal layer up to the surface of the skin, suprabasal, the keratinocytes change somewhat in phenotype as part of their normal life cycle. And so this basal layer here, one of the characteristics is they're proliferative, and they express a keratin called keratin 14, K14. That'll be important later. And then as they kind of move up, as the normal skin homeostasis is maintained, they lose K14 expression and they gain keratin 10 expression in the suprabasal layers. So they kind of have this change just during homeostatic processes, and they will also do that during wound healing. So when you get an injury to the skin, again, these basal keratinocytes will proliferate, they'll migrate to facilitate that closing over of the skin. And so they'll change in expression, and yeah, they proliferate, differentiate, and then they'll migrate into the wound. And so again, the question was, we know the skin is highly innervated. Other groups have shown that the free nerve endings, sensory nerve endings, actually contact keratinocytes directly, which is pretty cool. So we wanted to see again, kind of what is the relationship between nerves and keratinocytes, and how does that relate back to wound healing? So exactly there. So for this project, this was sort of like started in parallel with the iFlow plate work. And so for this one, we tried to go we went the conditioned medium route. And so we grew these DRG neurons, harvested their media, conditioned media, and then added that to keratinocyte culture. And so this was work done by a former student of mine, Avika, and with a current student, Alexander Noble, who's in the lab now. And so essentially what we did, like I said, is that we collected the sensory nerve conditioned media. So you'll see it abbreviated SNCM, collected that from cultured DRGs. And then we combined that with a pretty simple scratch wound assay. So this is a really common in vitro assay to look at wound healing of the keratinocytes. So you grow keratinocytes as a monolayer. You do a mechanical scratch. So we just use a pipette tip. And then we would add in our conditioned media or our control unconditioned media. And then you kind of observe the wound area closing and some other things. So we can see, does having this conditioned media from the nerves affect the wound closure and the keratinocyte function? So this is what the wound closure looks like under the microscope. So on the first row, we have our control media, so not conditioned. And the second row is our conditioned media. And so you take an image every day, every 24 hours for 3 days. So you can see at time 0, we got the scratch here. It's outlined in the teal color where the cells have been scratched away. So at 24 hours, 48 and 72, you can see in the control, the wound pretty much closes up. So the keratinocytes will actually migrate in and close it up. And we also control for proliferation in this assay. So we don't grow them in media that will have them proliferating too much to kind of decouple migration from proliferation. Because as you can imagine, they might proliferate and that could fill in the wound irrespective of the migration, but we kind of control for that. So you can quantify the wound area. So percent wound closure, this top graph here. So we did that. And we found that actually kind of contrary to what we expected, the sensory nerve conditioned media actually reduced the wound closure rate. So at 48 hours here, you can see the teal line is the conditioned media group. So the wound closure was around 60 or so percent versus in the control group, it was almost fully closed at 48 hours. And that was sort of, especially because our fibroblast work with the nerves we saw, that seemed to increase activation of the fibroblasts, which is known to be what you need for wound healing. And this, we sort of saw the opposite effect, right? We would have thought nerves are probably going to help, the keratinocytes migrate if they're pro-wound healing, but we saw the opposite here. And then this graph here on the bottom, it's the same data. You can just calculate over time, like the amount that the cells have migrated. So again, the keratinocytes in the sensory nerve conditioned media were slower. So the rate of migration was reduced. So we're like, okay, well, let's look at some more things here and maybe we can figure out what's going on. So one of the questions that we got was, well, is it just because having them in conditioned media makes the cells unhappy? Maybe there's just factors in there that they don't like, and so they're just not as happy, and that's why they're not migrating. And I will say we did some optimization experiments. So we don't add 100% conditioned media because, as you might imagine, if it's with the neurons, they're going to produce waste and whatnot and kind of use up the nutrients that are found in the media. So we actually do a 50-50 split of fresh media and conditioned media so that when it's added to the keratinocytes, they still have their nutrients that they need. But so we're like, yeah, that's a good idea. That's a good point. Let's see if just sort of the general cell viability is affected by the conditioned media. And so I'm not showing it here, but we also did kind of like a viability assay, found that there was no effect, no negative effect. And we also looked at mitochondrial activity. So we used MitoTracker, so a fluorescent dye that shows you mitochondrial activity. You can quantify this signal. So we did that. And we found at 24 hours and 48 hours, there was no difference between the groups. So the sensory nerve conditioned media did not seem to have a negative effect on mitochondrial activity. And then at 72 hours, kind of surprisingly, the cells grown in sensory nerve conditioned media had an increase in mitochondrial activity compared to the control. So they're migrating less. Their metabolism at least is not affected, maybe even a little bit boosted. So kind of interesting findings there. And then the last thing we kind of looked at, not the last thing, but the last thing I'm going to talk to you about today is we looked at the phenotype. So I mentioned to you that these basal cells express K14, keratin 14, and then as the wound closure happens, they'll kind of migrate up and express K10. Now, these cells that we use in culture are an immortalized keratinocyte line, and they're basal keratinocytes, so they should express K14 pretty much uniformly. But we again did our conditioned media experiment with the scratch, and then we quantified the expression of K10 and K14, so those two keratins. So you can see the images here. I should have said this for the other ones too, but this is essentially the wound edge that you're seeing there in the dotted line. So we're looking kind of to either side of the wound edge, and that's what we quantified. It's kind of, I think it was about 300 microns to either side of the wound edge. So you can see it closing up here. K10 in green, K14 in red. Most of them are K14 positive. We quantified this at our time points, 24 hours, 48, and 72. And we found that at 24 hours and 72 hours, so the two graphs on either side there was actually significantly more K14 positive cells with conditioned media. So when we cultured in the sensory nerve conditioned media, it seemed to kind of boost or retain this basal population more so than that suprabasal population. So there did seem to be a little bit of a phenotype shift there when we cultured with the conditioned media. So we did some work also looking at homeostatic conditions. So we didn't scratch the cells, we just kind of simply grew keratinocytes in a dish and repeated these conditioned media experiments. I'm not going to show you all of that data, but it was sort of interesting when we looked at the phenotype of homeostatic keratinocytes. So they hadn't been scratched, they were just growing in a dish as a monolayer, added the conditioned media on. There wasn't a change. So it seemed like potentially the conditioned media, the effect of the neurons, the nerves, might be dependent on kind of like what's happening to the keratinocytes. So when they're just in a dish with nothing happening to them, it doesn't maybe have the same effect as a wound healing scenario, which is sort of interesting to us, and I think makes a lot of sense when you think about kind of wound healing there in the microenvironment, all the things that are happening, switches turning on and off. You need the initial keratinocyte migration, proliferation, but you also need that to turn off at some point to go back to a quiescent state. So we're kind of thinking that nerves might have some sort of role in sort of maybe that switch back to quiescent from the active state. So to kind of summarize that, again, we're looking at the sensory nerves with the DRGs here. And so since we use these in this case, since we use conditioned media, we know it must be soluble factors found in the nerves are secreting. They're having this effect on the keratinocytes, where we're getting an increase in migration, but an increase in kind of this basal phenotype, and also potentially sort of the mitochondrial activity, so some sort of metabolism related effect. And we go more into that in the publication if you're interested in taking a look. So it's sort of, I think, a really interesting story that nerves are not just sort of always, pro wound healing, like go, activate. They might actually have a role in bringing back down to the quiescent levels that you need after you've kind of the wound's resolved at that stage. Pro healing factors. And so then the last thing we did in this study was we wanted to see what factors could they be secreting. And so no one has really, to my knowledge, done this before with these DRGs in culture. So we took our cultured DRGs, collected the media again, that conditioned media, and then we sent them away for mass spectrometry to do protein identification. And again, I'm not kind of showing you all the results here, but this is a heat map kind of summarizing some of the interesting proteins that were detected. So our control is just blank media, so you'd expect there to be nothing there. There were some keratins that were probably from skin or hair, so we took those out, human ones. They were taken out from both groups. But we did find a lot of proteins that are known to be involved in wound healing that we can now kind of follow up in subsequent studies. So things to do with kind of ECM remodeling, some collagens, MMPs, a few kind of regulators of inflammation. So some interesting targets that we're now following up. In particular, we're actually following up on this serpin A3N, which is a protease, which has a few different roles, known roles in wound healing. So it does seem like our nerves in our culture system do secrete a number of pro-healing factors. So just to tie it all back in again, this idea that what we're doing here, I talked to you about a lot of the in vitro side of things, but the idea is to kind of bring in that in vitro knowledge. Now we can translate that to some of our in vivo models, maybe go back and forth to the cell models, when it's more convenient or depending on the research question that we're asking. And we've got these kind of tools at our disposal and collaborators, which is awesome. So that's it for me. I'd just like to thank everyone for your attention today. Thank you again to Dr. Wiley and the organizers of this seminar for inviting me here today. Past members that did a lot of the work that I shared with you today and our collaborators, and I'd be happy to take any questions.
Ties together how to design something, figuring out what's needed. So people not in the modeling world, that's where I started my life. Even the model's too complex, they're useless, so you need to know what I'm doing. But you're making me scared now that I don't really need. Yeah, questions in the audience to start? Dave?
I have a really simple one. When you're setting these up at the IPO plate, why is the axon encouraged to go down into that middle well rather than just going off in the other directions?
That's a good question. So it's maybe a little hard to see from they do sprout axons in all directions for sure. I don't know if they just sort of, we have we coat it with laminin to make them happy, but there's no specific cues. So I think it's just sort of like if they can go that way, they'll keep going that way, but we will see sprouting kind of in all directions. And yeah, we've talked about maybe making it more directed, like introducing some sort of a gradient, growth factor gradient or something. And we haven't done it like fully followed up, but it did seem like if we actually seed in the fibroblasts kind of at the same time as the DRGs, that seems to help them want to grow that way. So probably the fibroblasts are secreting factors to attract the neurites as well. Yeah.
We'll also mention masked up really well. We show points taking your bar mask. I'm just wondering, however, there was that one particular bar map that showed the fold elevation of cytokines. I think it was each, we had a very large fold, like 6 or 7
Yeah.
right? I did notice the error bar was rather large, so for that one in particular, was any statistical significance testing better?
We did, yeah. Sorry, I thought it was a little bit easier. There we go. Yeah, we did. So nothing was statistically significant in this. It was sort of like a little more proof of concept that we are able to kind of like run these, harvest them in this way and kind of get this data set. And I think the DRGs, they're in all the things we use them for, they can be very variable. And I think it's just sort of like a nature of the biology, right? You're like, so our biological replicates are like from a mouse, right? We take the DRGs, that's what we consider like a biological replicate. And so I think it's just variability between animals, and we often do see that. And so, yeah, it's just, we saw that in our mass spec as well. Like it just kind of seems to be the nature of working with the DRGs. So yeah, we did test, but there was no statistical significance. Yeah.
Visually looked good, but was actually functional.
The limb, yeah.
Visually looked good.
Yeah. Oof. Trying to think if I want to say there's a few papers that have done a similar. They've done it in newts too. I want to say at least one of those, maybe not that one, but they have done like a sensation test. So they'll pinprick it and they'll see that the limb like responds. So functional in that way, maybe at least. Yeah, that's a good question. Yeah. Yeah, yeah.
For hypoplate, if I understood correctly, you're using mouse neurons and So, is there any issue in mixing like species of cells? Would it make sense to use? We're not using mouse
Yeah. Yeah, that's a good question. So it's not uncommon for people to mix because we've got that question a few times, and so it's a little hard to say. Ideally I would want to use everything human, kind of just for like the translatable power to that. But it's really hard to get a good population of peripheral neurons. Like you can differentiate them from iPS cells, but your amounts are going to be low, and long story short, we started out with the DRGs just because we could get those and use them easily to start out with. So I think in an ideal world, we would use both species. And we've talked about just getting some mouse fibroblasts, like primary ones, like taking them from the tissue and kind of repeating some of these experiments. And so that would answer the question, is there actual differences? I think they're fairly homologous. A lot of the ones we're sort of interested in, like the growth factors and whatnot. But yeah, I guess I don't really know the answer entirely to that. So yeah. Yeah, that's a great question. So I am afraid of the immune system because I find it very complex and I tend to sort of ignore it, but I think we're going to have to eventually incorporate it in some way, absolutely, because it is so essential. And we did see a lot of sort of immune related proteins when we ran the mass spec. And so I think for sure that's probably a component. And I think what we're kind of seeing with some of our other in vivo work and whatnot, I think the role of nerves is often pretty indirect. So I don't know, we sort of showed this direct effect, but I think they also operate on other cell types that then, affect the keratinocytes. So for sure, I think we're going to have to start thinking about other cell types and incorporating. Yeah. Ooh.
Because like all like in your control experiment was crashing again, or is it like everyone is then
Yeah, that's a good question. We kind of yeah, it was like a few hundred microns that we looked at, and we thought we might see, yeah, we thought we might see like along the wound edge, maybe like K10 there. Like if you're thinking about, in vivo, that's what you would get, right? Is kind of this, and we didn't really see that, so we haven't like fully quantified that, but just sort of visually, I know that wasn't really what we saw. It looked pretty uniform no matter where you looked at in the dish. Yeah, yeah.
So thank you so much for the interesting talk. I was wondering what kind of neurons are used in the IQ plates, and is it possible to use a neuronal cell networks?
Yeah, so we use the primary mouse dorsal root ganglia, so that's like the whole nerve that we put in the dish, and it for sure is possible to use others, but as I was kind of saying, like it's neurons are quite difficult to culture, and if you differentiate a stem cell type into neurons, then it's either like they're in that dish, they're kind of there. You can't really move them at that point. So there's a few kind of limitations that we haven't overcome. So yeah, for sure, there's different sources, but it's just sort of like picking the path of least resistance, maybe. Yeah, we haven't done that. I've thought about it. So it is the whole nerve. So it's even, and so we don't really know at this point, is it the neurons that are having this effect, or is it, there's lots of glial cells, right? Like Schwann cells and astrocytes that are associated. And so I'm sure those are secreting a lot of the factors that we're seeing, and immune cells are there too. So, um, I've gone off topic. Oh yeah, yeah. So I think for sure we could look at doing something. The iFlow plate is, the design's really nice because it's open top, so we could definitely look at some sort of stimulation of the neurons and see, does their firing matter in this system, which I think would be a really interesting question, because when you have a wound, you get like this hyperexcitability of the surrounding nerves. So yeah, great question. But they're sorry, what? Oh, Yeah, well, so I mean, when they're they're segregated physically from the nerve, so they don't, so it's just fibroblasts in that chamber. But yeah, I mean, they do for sure like proliferate more, which I showed with the nerve. So they do kind of yeah, we had to kind of optimize the density we started with and things like that, so they didn't overgrow because that's impossible to quantify everything and just yeah. So there was that aspect for sure. Yeah, so if we're translating to 3D and we use some sort of a hydrogel, we can mix in fibroblasts to be like the artificial dermis. And so yeah, I think we'll probably have to re-optimize what density that's at, and they're gonna possibly degrade the hydrogel over time. So we'll have to look at, yeah, probably gonna be a lot of variables for sure. Yeah, I think most people that kind of do incorporate nerves into biomaterials, they're like mixed in some way a lot of the time. I think so. We haven't started. We're like about to start these experiments to see, but I don't think. I find it hard to think that the nerves grown in the one chamber are going to extend axons through and then up into a gel. Like I don't see that really happening unless you have some like crazy cues that they. Yeah, like so that it's all embedded into one. Because I think they'll have trouble going from one substrate to another. So yeah, the other option would be to yeah mix them in and kind of see if they can spread through the. Mhm. Mhm. Yeah, I mean, there's a good body of work that has like encapsulates neurons in hydrogels and that. So I kind of would know from like they love hyaluron and like they like a softer gel and they love laminin. So there's kind of things we know that generally support them. So it's going to be adapting kind of what this Ang lab uses and how much can we kind of keep the same for consistency versus like hopefully we don't have to reinvent the wheel. But yeah, I think that like generally that's known like what neurons like in biomaterials, but yeah, it's sort of adapting the whole system. Hmm. Hmm. Yeah, it kind of brings us back to that compatibility issue. So I didn't present it here, but it turned out we were worried that that would be an issue, but it turns out the fibroblasts like the neural basal media that the neurons, they actually like the media that the neurons are grown in. So that saves us a lot of, and the keratinocytes as well like it, and the keratinocytes actually do even better in the neuron media than their own media, which is, I mean, maybe not too surprising because there's other factors in there, but so I think we'll be okay for that initially, but yeah, for sure that becomes an issue again, right? When you're mixing like various cell types into one construct. Yeah.